your team to accelerate your clinical transitions on the field of cell and gene therapy field. So I'm leading the clinical partnership opportunities in Asia for Sativa. Basically, our approach is starting from the basic research stage until the clinical transitions. So in between, there are scaling up process development processes that I'll be introducing later. But before I start, I would like to share with you please allow me to introduce who we are. Sativa is a global provider of technologies and services that aims to advance and accelerate the development of and manufacturing of therapeutic products. So our customers ranging from startup companies, pharmas, biotechs, manufacturing vaccines, or even monoclonal antibodies as a drug. So our job is to supply the tools and services that they need to work better, faster, and safer for the patients. As a trusted partner to our customers that range in scale and scope, Sativa brings efficiencies to the organizations, both in research and manufacturing workflows. This is to ensure that the development, manufacturing, and delivery of the products meets the regulatory requirements and be safe to the patient as well. Okay. The vision of Sativa is a world in which access to life-changing therapies transforms human health. And our mission is to advance and accelerate therapeutics. So before I start, I would like to share with you my understanding of the trends, challenges, and opportunities in the CGT field. As we all know, there are increasing number of clinical trials every year, and these trials are broadly categorized into 4 categories. One is gene therapy, one is gene-modified cell therapy. This is mostly widely approved now. I can't say widely approved, but a few. Hopefully, it will be widely approved later. But this is mainly focused on CAR T therapies and cell therapies as well. A major section in Asia, because a lot of companies and are doing mesenchymal stem cell-based therapies or somatics-based cell therapies. And the last one is tissue engineering. We can see that most of the trials are actually in phase 2, registered in US FDA or equivalent government authorities around the world. But the number of phase 3 studies are actually much higher than the previous years, which means that we'll be able to see more of these cell and gene therapy products entering into the commercial world. And the US FDA authorities also predicted that there will be about 20 cell and gene therapy products to be approved every year in 2025. And since there are increasing focus on indications, right? So the focus is really on big patient populations for considering the clinical and preclinical development in cardiovascular, central nervous system, and diabetes space, right? These are really the systematic diseases and affects a lot of the patients. So the first gene therapy product for critical limb ischemia is already approved in Japan. And for clinical trials in cardiovascular, related to cardiovascular or CNS indications include diabetes-related complications, Parkinson's disease, critical limb ischemia, stroke, peripheral artery diseases. So those systematic diseases are the focus at the current stage. And I mean, with so many clinical trials ongoing and so on, the development of CGT field is really fast. There are still a lot of challenges in developing those products. That includes translation of a lab process to commercial manufacturing. As we all know that the lab process is really manual. We started from culture dishes to BSC hood that that doesn't really require a very stringent environment to operate. But for commercial manufacturing, especially when you want to enter into clinical trials, the manufacturing space has to be clean rooms, a different grade of clean rooms, different processes that allows the product, the manufacturing process to be entered from one room to another. Under very stringent CGMP requirements. But the translational process from lab to commercial manufacturing it's not easy. You cannot change the product potency, but that actually always happens when you try to scale up from small scale, a few mils to hundreds of mils to liters or to hundreds of liters. The entire translational process is there's just too much variations, and the product potency may change. So that's very critical step, and that's one of the challenges in the field as well. And developing the manufacturing process that produce a high quality product with reasonable cost is really crucial, because currently we all know that the CAR T products are really expensive. Each dose costs about $500,000 US, and that's just, and that doesn't guarantee any profits for the companies that develop these products, because the cost of processing those cells and the R&Ds are just too expensive. And also flexibility of manufacturing platform to cater product specific parameters is another crucial factor. There are quite a number of platforms that we can use to manufacture the cell-based products, but some of these platforms are just too rigid that you cannot change any of their process or do some cell-specific parameter changes on this platform. So I would highlight that the flexibility of a manufacturing platform that is able to help you to translate your lab process to commercial manufacturing is really crucial. And lastly, human error is unavoidable. When we do research, we all know that there are human errors. You add a bit more of reagents, or when you transcribe your results from your computer to your notebook, there's a tendency of creating human errors as well. And that need to be minimized systematically, especially in commercial manufacturing, when your upstream process, you have one number wrong, that basically affects the entire product. So no one is the expert of everything. A production professional who runs the lab process may not be an expert in quality, understanding of GMP concepts. So collaboration is really the key to solve those problems. Especially within the organization, different departments have to talk to each other to resolve the problems together. And also externally, we need to work with different partners, suppliers, to try to minimize the errors that you will probably experience during the manufacturing process, and also in the research process as well. So this is the Global Biopharma Resilience Index. Singapore has ranked 8th globally on this index. And we can see that of the 5 pillars here, Singapore is quite good in supply chain resilience, manufacturing agility, government policy and regulations. But there are still things to be done to bring up our talent pool and R&D ecosystem. So in the later slides, I'll be focusing on these gaps and see how we can help Singapore as an organization, how we can help Singapore to improve the ecosystem and help to train more professionals, and to train more professionals to be able to expand the industry in Singapore. And so our CGT journey focus on deepening the capabilities and pursuing automation. And as also our initiative is really to help to develop local talents. And lastly, how we can expand the industry in Singapore through collaborations and partnerships. Just a brief history of Cytiva's cell and gene therapy. So we started from the GE Ventures initial investment in 2013, and eventually we have built collaborations with different partners like Mayo Clinic, CCRM in Canada. We developed a joint translational development partnership, and we did a few acquisitions as well. In 2016 and 2017, we have acquired BioSafe. BioSafe is the manufacturing of a lot of cell isolation products. Primarily, the CPAX has been used in cord blood industry across the globe. I think there are about 300 plus cord blood banks around the world, and Singapore has 3. And most of these are actually using the BioSafe CPAX to do the cell separations. And Asymptote is acquisition is done in 2017. It's a cryobiology. The Asymptote produce a lot of cryobiology products and cold chain logistics, so that enables Cytiva to have an end-to-end process. And in 2018, we have collaboration with CBMG and delivered our first end-to-end flex factory solution in China. As well as World Courier. We are working with World Courier on enhanced logistics to help our customers to bring their products from one region to another with GPS trackings and real-time monitoring of the temperatures of the products to ensure that when the sample is shipped from the manufacturing site to the clinical site, all these samples are within the validated range and safe to the patients. So GCON is another collaboration we have, Flex Factory. This is to manufacturing household POD infrastructure. So basically a whole set of GMP facility can be built through this GCON. And following that, the solutions for cell and gene therapies we have, basically we have a very broad portfolio of CGT technologies, consumables and reagents, including the cold chain logistics I just described. We have the ViaFreeze that is dry, using electricity to cool down the samples throughout the journey when you want to send the samples from a lab to clinical settings or vice versa, and with tracking. So and on the bottom left, you see enterprise solutions here that includes the CGT infrastructure, the Flex Factory, and we also have process development services, which is a fee-based service for CGT with full transparency and handover. So basically, your lab manufacturing process can be through a tech transfer to our fast track team. We'll be able to develop a clinical grade process. After tech transfer, you can just bring it back to your CGMT facility for production. For training and education, we do offer CGT courses for users, operators, as well as in-house maintenance of the products or the equipments used. And this, I'll share a bit more of the details on training of Cell T1 courses, which includes both classroom trainings as well as practical trainings. And automation solutions, all of our equipments actually can be automated, and we have pre-priority software for controlling individual CGT systems with third-party connectivity. So basically, your lab equipments can be connected to this digital system, Chronicle. So basically, all this data can be sent through your equipment once it's generated to your system, and this system helps you to do analysis as well as to generate EBMRs, ESOPs as well, and e-records. So all this very helpful when you bring this manufacturing data to the regulators, especially at your IND stage. That helps the regulators to have a deeper understanding of your manufacturing process that facilitates the approval of this of your products. And okay, so the capabilities of cell and gene therapy process development, our PD team, right? Our current focus on cell types are CAR-T, T cells, NK cells, ES or iPSC-derived cells, mesenchymal stem cells, and dendritic cells. And we have we have put this into upstream and downstream. So upstream, we have experimental designs, high throughput process developments, cell culture media screening, development, and transformation. So we have a new partner called Nucleus that we are able to help you to generate immediate ingredients that that may be most suitable for your cells, specific type of cells, and technology transfer. Viral, non-viral genetics is included in the PD service as well. And scale up or scale out, you may either choose one. Scale up can go up to 50 liters at this current stage for cell-based products. And scale out could be you have multiple bioreactors in the same site, like what Novartis is doing. And downstream, we have design of experiments as well. Cryopreservation, final formulation, formulate or fill improvements, including tech transfer. So upstream and downstream that puts all the entire manufacturing process together, starting from your sample collection, manufacturing, freezing, and also delivery or storage to delivery. And we also have analytical and other capabilities that includes assay developments that gives you the COAs of your process so that you can monitor the manufacturing process well. And we also have SOP developments, analytical transfer, tech translation, process closure, automation. So all these are actually enabling the transformation process of manual lab process, small scale to a large automated and digital manufacturing process. So we've been focusing on automation a lot. This is probably the last slide on automation. And so automation allows us to reduce and remove manual labor and touch points during manufacturing process. This is super important in a regulator's view, especially in terms of evaluating the performance of the manufacturing process and what are the trade-offs you can take. Just give you an example in blood processing industry. So in the lab, when you collect blood samples, you want to take out the buffer coat, right? You will do aliquot in 50 ml Falcon tubes. For if there's 1 liter blood you collected, basically you need to do 50 ml Falcon tubes manually in the centrifuge to spin it, to spin out, and then you collect the buffer coat, right? So that introduces a lot of risks in terms of contaminating your sample, and also a lot of labor requirements. You have to balance all these tubes in the centrifuge. But in terms of clinical manufacturing requirements, you have one simple device that can process the blood sample continuously, regardless it's 1 liter or 2 liters. You can just place the sample in bags, and then the equipment will just automatically separate your samples, your plasma, your buffer coat, and your red cells are in different chambers. And you can just collect whichever sample you would like to do downstream processing. And that actually reduced the time from probably a whole morning to just 1 to 2 hours. And this integrated and digitalized manufacturing process that gives you the ESOP, translates your manual SOPs from a paper or Word document to ESOPs in the system, and all your records in the lab to e-records. So BMR to e-BMR is really crucial when you do the regulatory submissions. And knowledge from bioprocessing, taking the advantage of our knowledge in the bioprocessing industry, standardization of platforms and processes is really crucial for this emerging field of cell and gene therapy as well. So automation is important, and automation allows you to increase your scalability from 50 mil Falcon tubes to liters of products in the bag, and increase consistency as well, because it's basically no human involved. You just need to press the buttons using the set protocols to process the samples, and everything is done through biosensors, through your predefined SOPs, protocols installed in the equipments, and that it decreases human errors. So you don't have to worry about noting down 1 million cells that is generated from your cell counter, and then you have to note it down and do your manual calculations on what will be the media that you have to add into your next stage and how you're going to aliquot all this. So that can be done digitally using the system, which decreases the human error, because all the equipments will just talk to themselves and get the numbers, and give you a solution for basically how many mil of media you have to add into the next stage. And this also helps you to build trends, because all this data is captured in the system, and then you can use all this data to analyze your manufacturing trends, especially in your early development process. That enables you to save a lot of cost, and also save a lot of time in process developments to accelerate the entire manufacturing process. So evaluate and integrate early is really, really important. The advantages include the tech transfer, so that when you do your tech transfer early, that enables your process to be completed early as well. So cost reduction. Labor is the largest contributor to CGT manufacturing. And this report shows that 30 to 70% of the CGT manufacturing cost actually is on labor. And that includes the manufacturing, the QA, QC, PM, SCPM, and facility management. So with automation, that reduces to up to 37%, and the major portion is actually on facility, as well as the manufacturing process, reagents as well. So direct labor reduction is almost 20% with automation. So that is not just on the number of professional trained technicians that you have to hire, is also on the quality side, because a lot of times that when a manufacturing process generated the error, the quality person will have to come in and do analysis together with the manufacturing staff, and that takes time, that consumes a lot of labor as well. So cost reduction on labor is through automation is really important. One is to automate the manufacturing process. The other is really to automate your quality system or data recording system. Okay, on talent development, because we have identified that Singapore, there's a lack of professionals in cell and gene therapy field in Singapore. So we actually developed a fast track training, we call it Cell T1. It's classroom-based training plus practical trainings. So these instructions within the cell therapy process and cell manufacturing under the GMP procedures. So everyone will be trained on the GMP procedure of manufacturing the cells through automation. And the cost is divided into upstream, sample collections, and cell expansion, processing expanded cells into large numbers, and downstream applications of cryopreserving your samples, storage of samples, and delivering the samples to clinics. So that also includes the practical sessions that you will be able to do hands-ons. So after the course, the trainees will be enabled to apply detailed theoretical cell therapy process knowledge to upstream cell expansion downstream applications, and able to identify bottlenecks and troubleshoot your specific process, because each of the cell manufacturing process is different. Even for the same CAR T, which we think that is similar, but for different media of different type of cars, that your manufacturing process could be different as well. And there's no one that knows your process better than yourself, right? So the manufacturing supplies will be able to support, but the key person to identify the bottlenecks and troubleshoot them will be yourself, right? So this is really important to know more of the entire process, so that you have a better life when you do the manufacturing of your cell products. And the perform industry standard techniques related to cell therapy manufacturing with emphasis on T-cell process. Our cell T1 is basically generated based on the CAR T process. So there will be a lot of procedures that is related to CAR T, but that is also an advantage because most of the current products, cell-based products approved by the FDA is CAR T, right? So that process will help you to understand the regulatory requirements on the GMP aspects. And also implement strategies used for process optimization and evaluation. So at the bottom, the very small words, right, are the overview of cell therapy workflow and cell types, tube welding, all those aseptic techniques, cell counting. So those are all included in the Cell T1 course. We have also built a lab in Science Park. So we call it CEL, Sativa Experience and Learning Lab. The purpose of building this lab is to support Singapore government's Industry 4.0 agenda to build research, digital innovation capabilities, and talent development. So we can use this facility to do trainings as well. And what we offer for this, in this facility, we offer 70 training programs, which is a platform for idea exchange and access of latest technologies. Our vision is to be a partner, an enabler for Singapore to be a global node of technology and innovation. The program highlights for in Cell is the training courses, of course, and support ranging from the following. We have bioprocessing equipments and workflows, cell and gene therapy workflows, research and digital automation flows in all equipped in this lab. So if you are interested, you can scan this QR code for a visit. So industry collaborations is really crucial, as I identified early in the slides. We really need to collaborate with each other and build partnerships to bring the different sectors of the cell and gene therapies into the ecosystem. And Sativa is the manufacturing of these platforms, like to work with CDMOs, to work with clinics and researchers to link the entire ecosystem together, and then so that we can bring therapies to patients early. And so my role is on the clinical partnerships, right? But clinical partnership doesn't limit into clinical trials. We started on early research stage. As long as you have a potential cell therapy products with a prototype, we can work together on partnerships to understand your potential clinical indications, your product pipelines, what will be the regulatory framework that you have to follow, what are the CMC files that you have to submit, and for your IND or commercial roadmaps. We also can develop and execute initiatives to support those activities related to translational, preclinical, clinical developments, and basically foster collaborations between biotech and CDMOs related to the clinical product as production is needed. And our aim is really to build long-lasting relationships rather than just transactional working on 1 or 2 projects. So that's not the goal. Our goal is a lasting relationship and ensure that the strategic focus of Cytiva is aligned to your CGT program through deep understanding of the strategy and challenges in the industry and across different customers. We also try to develop creative solutions and offerings to enable both customers and Cytiva's long-term success. So we do have custom-made products for different organizations because each cell therapy product is unique and the current parameters may not be suitable. So we can do customized designs to work better with you. So this is just one of the case studies on CAR-T. Jointly worked with CCRM, development of a modular, semi-automated and closed T-cell manufacturing strategy. So this is the most this is also the most widely used T-cell manufacturing product approved by US FDA. But if you are interested, because there's time constraint, if you are interested, I can just share with you offline. So to sum up, what is your next step in your cell and gene therapy journey? Cytiva has the foundation, knowledge to help commercialize your therapeutic products. We have very deep understanding of the field, and we have expertise in critical areas of cell and gene therapy ecosystem. We have integrated solutions to help bring your therapy safely and effectively to the market. That's the end of my presentation. Thanks.
Thanks so much, Ming. So we have a couple of questions from the audience as well. So let me just ask on the first one. From one of our audience, Ming would like to really find out what are one of these process development capabilities that you've talked about. What are they offered in Singapore?
So our cell T1 course from Fast Track is not currently offered in Singapore, but the cell facility I've mentioned in Science Park has the entire workflow. So basically, when you visit the cell facility, you will be able to kind of understand the entire process that can be integrated into the cell manufacturing process you desire. But for the actual cell T1 course, we do have a few locations around the region. We just conducted one session in Shanghai, Shanghai Fast Track Center, first week of March. And also there's another one in Japan. We're also planning one in Korea. That's for Asia. For US and Europe, we do have Cambridge and Marlborough, 2 locations offering the course. But basically, the course is not just offered to Cytiva staff, it's not internal, it's external facing. So if anyone is interested to understand a bit more of the cell manufacturing process through automation or digitalization, you can sign up for this program. It's listed on our website as well. So if you just search Fast Track Cell T1 training Cytiva using all these keywords, you'll be able to locate the locations and the time this program is offered. But yeah, it's not just for Cytiva, but during the actual course, it will be a mixture of Cytiva staff as well as biotech companies or even researchers, because this really gives you an overview of the entire manufacturing process from start to end that is able to be applied later. Yeah.
Yeah, that sounds very exciting. And I guess this also answers Marin's other question. So the fast track training, it's available to anyone, so do sign up. Whoever's interested, it sounds like a really good opportunity to learn from some of the experts. I guess the other question, it's also, I know you touched a little bit about it, that at Singapore now in general moving towards manufacturing 4.0. So how important is digitalization actually as a part of a company's overall strategy to stay relevant or at the cutting edge of science? Specifically, maybe you can talk about how digitalization really has changed the cell manufacturing process as we see it.
I would say digitalization. I mean, it's interlinked, automation and digitalization, because if you don't automate your system, it's very hard to achieve the goal of digitalization, because if everything is manual and the process is conducted by a lab technician, everything is recorded on paper, and so that's going to be very difficult to digitalize, because
Right.
now in our lab we have flow cytometries. When you generate your flow data, you can print it in paper, or you can just save the FCS files, and you have all the numbers in your computer. But that's not entirely digitalization, because you still need to do the calculations in your probably with the Excel sheet or calculator. But with digitalization, right, if you are able to link all your equipments in the lab together in one lab monitoring system or lab management system, that actually helps you to pull all this information from different manufacturing processes or your analytical instruments in one big system. And all these systems are interlinked, right? So for example, if you just finish a cell count,
Right.
and you also did a CD34 flow cytometry, that the percentage of your CD34 in, and the cell counts. So the system will help you to do the calculation on the exact absolute number of CD34 cells in your manufacturing process. And that if you want to expand the cells, you'll be based on this cell number, you'll be able to the system, the digital system embedded SOP or the formulas will tell you how many mule or liters of culture media you have to add into your next stage. And this entire process is, if it's digitalized and automated already, right, you don't have to use your calculators.
Right.
And so that's going to be super helpful, because when you enter into a manufacturing process, manual calculations can be tedious, and you have to train different technicians. You need one person to do the calculation and another person to verify the calculation, so that you can move into the next steps. But with digitalization, everything goes just right to the point, right? And if assuming there's an error when you do the calculations, you accidentally entered the number wrongly, maybe just a simple dot, but that would cause significant downstream time consumed in verifying, or even releasing the products as not manufactured under your approved SOP, right? So there's enormous time going to be involved if there's a human error involved, and so that's just that's just too much time and effort, and costly as well. Yeah.
So in the interest of time, I'm going to move on to the next talk, but we'll invite Ming back again later for a combined session. So if you guys have any more questions, do feel free to keep asking them. So for the next part, we have from Paul Biotech with Dr. Zhang to share with us more about some of the available technologies to enable viral gene therapies. Dr. Zhang Peiti is responsible for the technical engagement with customers, the gene therapy community across Asia Pacific. He has over 15 years of experience in biotech research, development, and commercial manufacturing. So prior to joining Paul, he's held various roles in MSAT, biopharmaceutical industry. He was with A-Star. He was also a young assistant professor at NUS. So basically, it's a whole wealth of experience from both academia and industry. So without further ado, Dr. Zhang, you can take it away.
Yeah, thanks, Hunting, for the nice introduction. Just to confirm, you can see my presentation in full screen mode, right?
Yeah.
All right. So first of all, I would like to thank Biotech Connection Singapore for this opportunity to present our thoughts to you, the audience, and appreciate your time. Again, as Ming said, this is Friday, supposed to be take it slow, right? So I appreciate you are staying with us to hear what we have in mind in terms of looking into the future of cell and gene therapy. Thanks, Hunting, for the facilitation. That's really smooth. Thank you very much. So just now in the first talk, in Ming's part, you heard about his sharing on the big topics, right? So actually the global market view. So we actually have seen, how the market dynamics looks like, the global clinical pipeline. So you see the numbers, which is really convincing to propel cell and gene therapy as a whole new modality area into the future for the benefits of patients. We have seen really proposals in terms of manufacturing facility, equipment, specific technologies, training programs altogether, and heavy emphasis on digitalization enabled by automation. So I think, give credit to Ming really, because that really makes my part of introduction much easier. And what I decided to do is really to trickle down one level deeper to specifically walk through with you in terms of in the specific area of viral gene therapy, and to take a process developer perspective to let you hear from Paul Corporation, what is our perspective, what is our actually solutions, right? So this is not really meant to be a commercial talk. This is really a technical sharing for the audience to hopefully that you can walk away with some learnings in terms of accelerating, fast-tracking your viral gene therapy process developments. So before I jump into the technical part, again, just to put our organizations into the context. So Cytiva, Paul Life Sciences, we are both under the life science platform under Danaher Corporation. So we are actually two independent operating companies collaborating together very closely in the broad bioprocessing industry. And also under the same platform, you can see familiar names on the analytical side like SciEx, Beckman, Leica, Molecular Devices. Many of the tools you have already familiarized yourselves with. You have heard about it or even used about it for cell and gene therapy analysis. Of course, parallel we have diagnostic business, the environmental applied solutions operating companies all under Danaher umbrella. So if we walk along the drug development life cycle, so from hit to lead, this is what we typically call, starting all the way from target validation to final commercial supply of the drug substance and drug products. You can actually see, the positioning of our different operating companies within Danaher on this map. So clearly, Paul and Cytiva together, we actually serve our customers starting from your process development phase and supporting you all the way to commercial supply of your products. Now back to viral gene therapy. So just to set the stage, I think most of you in the audience know, broadly, we are actually looking at two categories: the ex vivo gene therapy, where we introduce the gene of interest using typically a viral vector to the cells outside of the patient body before we infuse back the cells into the patient, right? So this is also known as gene-modified cell therapy, which was very nicely covered by Ming just now. And also not forget to mention the other category, in vivo gene therapy, where we actually introduce the gene of interest directly to the patient body through a potent, effective vector, typically a viral vector. And if you really look at the common types of viruses that we use for gene therapy, we see retrovirus, including lentivirus for ex vivo gene therapy. We see AAV and perhaps more on the legacy side, adenovirus for in vivo gene delivery. And of course, one of the really rising stars is the oncolytic viruses like herpes simplex viruses for lysis of the tumor cells. So really, I think suffice to say, these viruses are really diverse in terms of physical, chemical, and biological properties. And also the gene packaging capacity of each virus type is also quite different. And if you really look at the market share, I think reasonably, you can see AAV lentivirus are representing the majority. They're really taking a huge chunk of the whole market share. So most of my technology solution sharing later on will be generally centered around the production of AAV lentivirus. However, we do have demonstrated cases that very similar technologies can be used either directly or adapted to the production of other virus types. So let's keep this conversation ongoing even after this discussion. Let us know your specific use case. I'm more than happy to provide technical consultation to you. So just to narrow down to AAV. So when we look at how the industries actually tackle the production, so let's start with the expression systems. Broadly, we are actually seeing these 4 major types of expression systems. The first 2, I think most of the audience would be very familiar. These are basically the triple transfection strategy, where we kind of divide all those genes of interest, replication, machinery, and helper functions into 3 plasmid, and through a transient transfection to deliver them to typically HEC293 cells, which are cultured either on flash surface or adherent culture, or grown in suspension, in shake flasks or bioreactors. And also not forget to mention AAVs can be produced using alternative expression systems like using baculovirus to infect typically insect cells like SF9. And in certain cases, adenovirus can be also used in lieu of the helper plasmid to ensure very high productivity of AAV production, especially in HeLa cells. So really, there's really no predominant platform. I can only say the triple transfection of HEC293 is being the more commonly used platform for AAV production. There are also challenges and a point to consider before you embark on your process development journey. After you have chosen your expression system, really, this is a matter of quality by design. If you look at the latter 2 expression systems, so just a specific example, if you decide to use virus to produce AAV, which is another type of virus, you need to have dedicated virus removal steps to take care of baculovirus and adenovirus, which would be deemed as process-related impurity for dramatic safety concern. So let me move on to the next slide. So this is basically a map of how viral vectors, especially AAVs, are manufactured in the academic lab. So I think for people who have actually worked in the R&D lab for in a vector core or similar facility would actually find this very familiar. Typically, we culture HEC293 cells in T-flasks or scale them up slightly to multi-trace, and then we perform the triple transfection using 3 types of plasmid. Give it a period of time for the viral vectors to be expressed, either secreted into the supernatant or accumulated inside the cells. This is followed by recovery of the viral vectors. Typically, we use detergent to solubilize the cells to retrieve the viral vectors, or we perform a freeze thaw to lyse the cells. To purify in the academic lab, the most commonly used workhorse is really the ultracentrifugation. So by exploiting the fine difference in the density, we enrich the viral vectors, the viral particles, to separate them away from other impurities. And this is followed by the use of labwares for formulation, before we put the material to non-human use, like proof of concept in vitro studies or animal models. If you really look at this map and look at the future need, which is really large-scale, cost-effective production of viral vectors for patient supply, there are actually a few major gaps. So definitely this process is hardly amenable to scale up. Basically, you have to scale out in a way, duplicating the number of cell culture vessels, duplicating the set of ultracentrifuge machines that you have. And this is highly labor intensive. You perhaps need to multiply by the same number of FTEs to cope with the high production demand, right? And definitely there will be multiple steps which are actually operated openly. So when you present this whole manufacturing process description to the regulator, there will be question asked in terms of the control strategy for adventitious agents, for example. So definitely these are not really viable solutions to commercialize a viral vector production. What are really needed? So when we look at the productivity, definitely, I think this is still a very hot, ongoing research area in industry and also in the academic facilities. Novel expression systems. So how can we produce more viral vectors per cell, right? Using the limited cell culture areas or volume. What are the scalable manufacturing technologies which are amenable to closed operation, to be automated, better controlled, that can really afford a process scale up and better control, leading to a consistent, reproducible viral vector process in order to ensure the batch success rate and also to control the manufacturing cost of goods. So when we ask this question, actually, sometimes it's actually worth looking back to see where we have actually covered, historically. So definitely, suffice to say, there are actually a lot of good learnings that we can leverage when we look at the map space, which also evolved for more than 2 decades to the level of maturity as we have right now. So for monoclonal antibody production, as we know, this is pretty much a very highly platform industrialized modality. We talk about seed train using various reaction vessels, put it to a sterile manufacturing in single-use or stainless steel bioreactors, harvested and achieve solid liquid separation using centrifugation with or without additional depth filtration step. And then, as you know very well, this is followed by typically a 3-step chromatography purification strategy with filtration technologies in between to buffer exchange, reduce the bioburden, and also at the end to make sure you end up with sterile formulated bulk. So accordingly, we can leverage on the same design philosophy to implement it to viral vector production for AAV and LV, and also not forget to mention other viral vector types as well. So if you look at the maps, the middle and the column on the right, so again, it's the same design philosophy. Try to scale up the capacity using single-use bioreactors to ensure better product changeover, from a cleaning validation point of view, and then achieve solid liquid separation using depth filtration steps. Make use of the combination of chromatography and filtration technologies to ensure product recovery, polishing, and impurity removal, for example, bioburden, virus removal altogether. So this is basically a kind of a visual representation of the process adaptation from the academic process, which I showed you earlier, to industrializable process that can be highly amendable to scale up to meet the future demand. So again, as I mentioned earlier, so changing it to a very space-consuming, labor-intensive multi-tray process to intensified adherent bioreactor process in our specific proposal, the iCELLis technology, and changing precipitation-based product recovery to depth filtration, changing the ultracentrifugation, which is really batch in nature and very small scale in nature, to more continuous flow-based technology, like the use of anion exchange chromatography, like Mustang Q for product polishing, ending with the use of TFF for formulation and sterile filtration to ensure sterility or low bioburden control for your drug products. So that's really the current status in the academic and the future state for clinical and large-scale manufacturing. But before we talk specifically at each unit operation or the overall manufacturing technologies, actually there's a note of caution, right? So this is actually something which is different from the map world, where typically we need 1,000 liter order of magnitude bioreactor volume to generate kilograms of monoclonal antibody products. In the viral vector world, the scale is actually highly diverse. So let me, you can see this as an example. When we look at the manufacturing capacity that's needed, or scale in this case, there are two factors contributing to this equation, right? So first of all, you have to consider the dose level, and this is highly indication dependent. We have really seen, for example, ophthalmic disease, eye diseases, where the viral vector per dose is really on the low end. And however, conversely, if you look at some of the high dose indications like hemophilia, we have already seen more than 1,000-fold or 10,000-fold order of magnitude difference between these two indications. The other factor is really the addressable market, your patient population. There could be rare diseases being exploited by AAV therapy. And on the other hand, hemophilia, DMD, muscular dystrophy, for example, these are really diseases with a huge population base per year, if you look at the incident rate altogether. So multiplying these two factors together, you potentially end up with 9 log. So 9 order of magnitude range of scale that's required. And now if you think about it, whether you are a platform company being a CDMO or a drug developer, you need to really consider what actually are going to come to your pipeline, and potentially it's also going to be a wide range of skill that's required for your manufacturing technology to be able to cope with. So with that in mind, let's now look at the process development journey. So as I mentioned, in the academic lab, there's a certain way of manufacturing the viral vectors, and there's a different end state in the industry where we need to consider the 9 log order of magnitude difference in terms of the manufacturing skill. So how do we bridge these two together? So it's always very easy to talk about process scalability, but what does it really mean to process development and manufacturing operations or executions? This is how Paul actually looks at process transfer and scale up. We actually divided it into 3 high-level process steps. The first is actually to perform a technical transfer. So a lot of knowledge transfer with the process originator side, being it a viral vector core, clinical production center, or academic lab, to understand the process parameters, where it's actually coming from, and to replicate this process in a process PD or even preclinical production facility to make sure you completely understand this process. You are able to replicate this process to the same in terms of productivity and yield as was reported earlier. And from there, to perform a proof of concept study to bridge, as I showed you earlier, the 2 process trains, the academic train and the industrializable process train, to start proof the concept that the same kind of cell cultivation strategy, whether it's adherent, if you change it from a T-flask to a fixed bed bioreactor like iCELLis, you can still achieve similar order of magnitude productivity. And by changing ultracentrifugation to, for example, anion exchange chromatography, you can also achieve comparable impurity removal, capsid enrichment, for example. And then into the second step of the overall process, that's where you start the true blue process development, where you start to really look at the design space, taking a quality by design approach, and from there to optimize the process parameters and to understand the process risk, right? And a very critical deliverable, we think, during the process development is really to improve the manufacturability. As we know, the manufacturing cost is actually a significant cost driver for the overall viral vector price tag, right? So over here, a lot of efforts should be really spent on improving the process manufacturability, robustness, process economy altogether. And then the end stage is really to scale it up. So basically, we typically do a 3-batch consistency run to make sure that we can successfully scale up a process, for example, from, let's say, 3-liter benchtop suspension bioreactor to a 500-liter large-scale bioreactor with the corresponding linearly scaled downstream train. And of course, this all ends with a tech transfer to the future manufacturing facility, whether it's our customer or the customer's chosen CDMO facility. So over here, we also feel that it's actually very important to really transfer the knowledge altogether with, as you know, the tech transfer documentation, the BOM, the process description altogether. So that is where the deep insights, the deep knowledge, know-how of the manufacturing facility, the equipment, consumables is actually very crucial for the success of the successful tech transfer. So just now I mentioned, so like two commonly used technologies for the triple transfection. On one hand is the adherent platform, on the other hand is suspension. I think suffice to say, this is often confusing enough for a lot of the end users, including our customers. So when you are at a point where you decide to scale up your or industrialize your manufacturing process, do you go adherent? Do you go suspension? How do you make a decision, right? So this is often a critical decision point. So this is how we look at it. There's really no one-size-fits-all kind of solution. It really depends on what is the selection criteria from the drug developer. So for example, we definitely have seen tremendous advantageous features from the adherent bioreactor. For example, ICLS 500, which is a large-scale fixed-bed bioreactor. In terms of the speed, meaning the speed to quickly develop the process, thanks to our very deep experience accumulated over the years, the speed to the market, so that you don't have to really adapt yourselves from T-flask to a suspension culture, and also the regulatory agency familiarization, right? So they have actually approved. Later, I'm going to flash a case study to you. They have seen this technology, they are comfortable with it. So from a regulatory point of view, it's actually easier for the regulator to approve, to receive and accept a process that's actually based on the same technology adherence. On the flip side, suspension technologies has its own advantageous features as well. As we all know, many of us actually, build up our career in the bioprocessing in the mAb world, right? So we know a lot about suspension bioreactors in terms of, let's just toss a few operating parameters like the gassing strategy, the scale up strategy, the seeding. Many of these are actually the advantages of the suspension bioreactor in terms of the ease to scale up. Pretty much you do a volumetric split in order to scale up from A-1 to end stage, right? And there are also a large number of CDMOs with already installed suspension bioreactor, which they can quickly repurpose from mAb production to viral vector production. So really no one size fits all. It really depends on what are really the key goals the drug developer organization as an organization are really focused on. And that will dictate really the selection of the technology. So how about the downstream side? I don't intend to really underestimate the technical challenge on the downstream side. Arguably on the downstream, for many of us from the process sciences side, it's suffice to say to a large extent on the downstream side, things are a lot more linearly scalable and predictable. So really the key for the downstream is really the selection of technology that are truly proven to be linearly scalable. So for example, the choice of your depth filter, it has to be consistent per volume or per area, regardless of whether you perform the purification or the separation from the lab scale through pilot scale all the way to the clinical manufacturing scale, right? The same goes with chromatography and TFF technologies altogether. So taking everything together, the choice of upstream production platforms, the choice of linearly scalable downstream technologies. I'm actually showing you one rendition of a platform AAV production platform process. So this is actually an adherent process that we proposed based on a lot of internal data, our field experience that we generalize to really achievable in terms of productivity, the process design, the sequence, and the specific technology that is actually shown over here. So suffice to say, we start with biothaw, cell expansion using various cell cultivation vessels like T-flask, multi-trace, or even expansion bioreactor from Paul, put it to main stage for cell seeding. So in this case, you are seeing iCELLis 500, which is a large-scale fixed bed bioreactor. And this is followed by, as I mentioned, scalable technologies like depth filter, TFF, bioburden reduction, filtration, chromatography steps to capture the virus particles and to polish it further. For example, to remove the DNA further, to remove the whole cell proteins, to remove the empty capsids altogether before we formulate it. Again, using TFF technologies, putting it through sterile filtration so that we end up with sterile formulated bulk before we perform the formulation, the final fill finish packaging altogether. So from here, you can already see using one such scalable, large-scale manufacturing technology, we can generate huge amounts of viral particles. In this case, order of magnitude 10 to the 16th vector genome per batch, even after purification. So you can already see, correlating to what I shared in terms of the scale that's required. And this is already sufficient for vast majority of the indications already. So really, it's actually achievable. It's actually transferable from academic to industrializable process, and there are already commercially available solutions out there. So I'm going to end my talk with two specific case studies. Otherwise, it will be just proposals. So in the first study, this is really a result of the collaboration between Paul and Avaxis, so later acquired by Novartis, known as Novartis Gene Therapy. So from the timeline, the Gantt chart, without much mentioning, you can already see, we started the collaboration in 2016, and it actually took less than a year from the Paul process development team to successfully develop the process on the benchtop fixed bed bioreactor before we successfully implemented to manufacturing scale iCELLis 500, leading to, as you know, successful drug approval. So this is really a specific case of Zolgensma. This was actually reported in the ASGCT meeting about 2 years ago. So if you're interested, you can log on to this website. By the way, this is really our true professional society if you're interested in cell and gene therapy. The second example is not really true gene therapy, but it's also about the production of viral vector. It's just that in this case, it's actually a virus for COVID-19. It's actually a vaccine as the final drug product. So as many of you know, so in the US, there was this Warp Speed initiative. In the UK, the government actually initiated a huge collaboration work involving therapeutic company like AstraZeneca, suppliers like Paul, and CDMO companies to work together with the kind of ultimate challenge of how to churn out a vaccine at light speed, if I don't call it warp speed. And this was actually achieved by Paul Corporation, of course, in collaboration with our past partners. So pretty much 8 weeks is all it takes from us to receive the lab scale process to successfully scaling it up to final manufacturing scale and successfully tech transferred to CDMO. So this technology is already successfully implemented in many countries in the world, I think to 2,000 liter bioreactor. So you can already see that level of, I can't echo enough of what Mingming shared, the strong partnership between supplier and the drug developer to make sure that we can really deliver a therapeutic or even a prophylactic to the market at unprecedented speed. So that is really thanks to the deep knowledge that really acquired, accumulated on the supplier side. So with that, I'm going to just end my talk to just, again, 3 key messages. If you forget everything of what I say today, really, the challenge is really to balance the speed to the market, which is the holy grail of everybody, to decrease the cost and also to decrease the risk right? How do we do it? We have to leverage on platform technologies using scalable solutions to make sure we reduce the risk. We also reduce the cost by intensifying the process. For example, using Paul's fixed bed bioreactor, we can actually reduce the manufacturing footprint by as much as 2/3. So that's actually, if you think from a clean room validation maintenance point of view, that's going to be tremendous cost reduction. And also suffice to say the speed, right? Just recall the case that I showed you in terms of the AVAXis. So less than 1 year, that's what it takes. 8 weeks is what it takes to scale up a vaccine, viral vaccine process development. So with that, I'm actually sharing with you my email address over here. So again, never intended to be a commercial talk. This is really our sharing of how we perceive are the strategies to accelerate cell and gene therapy, especially the viral gene therapy. If you have any questions, don't hesitate to write me. So let's continue the conversation from. Thank you.
Thank you so much, Niti. So it's really exciting to hear about all this and experience. And what I really enjoyed was also where you talked about the processes in the academic lab as well as the processes versus at manufacturing scale. So I was kind of curious. Given that now we're trying to really accelerate and move processes from just on-paper concepts, proof of concepts in lab, and we know that drug development is a really long time. And failure rate is so high. So how do you actually know when's the right time to move away from the academic process and actually convert and do the tech transfer from the academic process to a more of an industrial process?
That's a good question. So I think there are two thoughts. The first conventional thought, if you do a stepwise drug development, right? So I think everybody should know. So, when we talk about drug development, typically we start with target validation, we start screening the compounds. In the case of AAV, you start selecting or engineering the capsid protein to make sure you're actually targeting the tissue that you want. There's actually minimal tissue crossover, it's actually well tolerated before you finalize on the drug candidate. And that is usually in the conventional way where you put this asset to the process development journey, right? So I think in the conventional thought, that should be the outset where you start really evaluating how to adapt your academic process to an industrial process to be scalable and acceptable by the regulator. Now, the other thing, we're in an increasingly competitive landscape. So I think you still remember, or at least are impressed by the huge numbers presented in Mingming's presentation. A lot of competitions, I think this is also increasingly the case even in our red dot, right? We are seeing a lot of startup companies sprouting in Singapore even. So the key is, do you want to go conventional and risk of somebody else just scooping your idea, right? The targets are really limited, right? So if you look at PD-1, how many companies are working on PD-1, right? Globally, hundreds of them. In China, more than 100, right? So how many can eventually make it to the market to be still commercially available, viable, right? So I think in the new school of thoughts, and this is also kind of resonating with me, is we need to also consider parallelizing a lot of these workflows instead of doing it stepwise. But it also represents substantial risk, right? If you prepone your process development, you are actually exposed to higher risk where your variants or your candidate actually failed your talks or your preclinical evaluation, right? How do we mitigate such risk? So I think this is really beyond the PD topic, but I worked on both the pharma side and CDMO. So I think it really again boils down to embracing platform technology as much as possible. So regardless of the subtle difference in the variants, your process is actually to a much lesser extent going to be impacted by the change of the drug candidate, right? So for example, still the use of this bioreactor technology, still use of the same purification train with high level, very similar process parameters that can be applied from asset to asset. So I think the embracing of the use of scalable platform technology, that should be really central if we really embrace the new thought.
Yeah, and going back to what you just kind of mentioned, talking about platform technologies and also like really the increasing number of startups within Singapore. So, and with BCS, we do have a lot of people who are working in startups. So I was just kind of wondering, at the start, you mentioned 4 different dominant platforms, the adherent and suspension cell options. So if you're a startup, what would you consider when you're trying to pick, with this huge array of platforms, what would you consider to be able to choose the right platform for your company, especially for startups?
So pretty much this is a strategy question, right? So you have to really look at your internal factors. You have to also look at externally. You have to do some competitive analysis to differentiate your positioning of the drug, right? So internally, look at what capabilities or what material you already have, right? Be it a cell line or experience with a particular expression system. For example, if your company is actually formed by virologists who are very confident, comfortable with molecular biology design in terms of the adenovirus vectors in the background of either HeLa or HEC293. So why not go ahead to exploit that route, right? And also look at internally. If I choose that route, what is the impact to my further drug development journey? So for example, as I mentioned, the use of adenovirus for AAV production. So how confident are you in terms of adenovirus removal, right? So, I mean, it's feasible. It's not like a rocket science, so to speak, but you need to put all those into the context to say that, hey, I want to produce my AAV using this platform. That means I would actually spend plus 2 months on, the viral filter study, plus another 2 months for back and forth discussion with the regulator to see whether they are comfortable with our control strategy, taken together, resulting in plus 6 months as compared to triple transfection. What is the patience of my investor, right? What is the exit strategy and timing that they are looking at? And how much, how does my cash flow actually look at, right? Yeah. And I don't have that GMP manufacturing facility. Can I find an external CDMO who can deal with adenovirus HeLa cells? Can I bring HeLa cells, for example, to China? That's a challenge, right? So I think put everything together, look internally, what capabilities, externally, what's available, what's actually on the competition. From there, pretty much it's a strategy question. Yeah.
Thank you so much. We're going to move on to the last part. I'm going to invite Ming to join our panel, and then we'll have a bigger discussion as well. We talk a lot about cell and gene therapy, and it sounds like the world has been doing this for a bit longer than in Asia, and it's relatively new in Asia. So maybe each of you all can share a little bit of your thoughts. It's rapidly growing in Singapore, but what is it you think is needed now in Singapore, in Asia, in this region to build the critical mass needed for these modalities in the region? Is there a manpower issue, a facility issue, or maybe regulatory issue for it to really take off in this region?
Maybe I will start. My understanding, based on years of experience in Singapore, is that we are short of facilities. We really don't have a facility or good CDMO in Singapore that is able to have the capacity to support so many startup companies to manufacture the cells, right? And also to do the process developments to scale up. And so that's one of the key issue. But looking forward to actuaries that is going to be built up at Outram Park, right? So that's going to solve some of these problems. And in terms of manpower, that's I mean, comparatively to the rest of the Southeast Asian countries, well, they are quite good already. We have very good talents in Singapore in the biopharma field. But in terms of cell and gene therapy, because it's very specific field, and we always say that your process is your product. So basically, if you change any part of your process, your product will change as well. Especially for cells, you change one growth factor, that your end product would be a different subtype of cells. That could be still I mean, based on a few biomarker analysis, you can still say, oh, this is the type of cell I want. But if you do more genetic studies, you'll see that it could be a subpopulation already. So with all those those things that you have to consider, right? We have to really train up a good group of professionals to operate in the cell therapy field, especially in the production field, as well as quality is very crucial in terms of cell manufacturing process, right? Because comparing to the pharmaceutical industry, that small molecule drugs, right? You can do your sterility. You can clean up the compounds later at a very later stage. But for cells, you can't autoclave the cells, so you can't extrude the cells. So your entire process has to be GMP compliance. You have to be very careful at the beginning when your sample is collected at the starting material until the very end, after 1 or 2 months of cell manufacturing, generate enough cells for therapeutic dose, right? So the entire process has to be sterile, and the regulatory environment is getting better now. In the past 2 years, HSA has come up with quite a number of guidelines, and so that's very helpful, especially in the recent years, that we know where we are going in the regulatory framework. And so, I mean, we are short of facilities. The problem is going to be solved soon in the next 1 or 2 years time. And talents, I mean, we have so many talents in the pool that we can just quickly train them up. Especially when we have good GMP facilities, we can train those talents in the facility as well. And regulatory framework is getting easier. So I guess that there's no doubt Singapore is going to advance in the cell and gene therapy field in the next couple of years. Yeah.
What about you, KT? Do you think there's anything stopping us from really delivering more gene therapies in Singapore?
So I think, first of all, this is actually all trending very promising. If you look at it, I think first thing that really came to my mind is we have to be really patient, because drug development is actually a high risk and also a very long journey, right? If you look at it, we're talking about a lot of acceleration, warp speed, and altogether, but it's still taking years, right? You know, you can do kind of a conditional approval based on phase 2 data, but you still have to do the phase 3, and there's still a chance of failure, right? So I think globally, well, broadly, if we look at Singapore, I do see we have many of the boxes already checked. We started I think led by A*STAR and also like EDB, so the Biomedical Sciences Initiative, that actually started, I think, more than about 30 years ago. This is better than me. So definitely we had already trained up a critical mass of biomedical researchers covering different spectrum of the whole value chain, from discovery, even fundamental research, to disease models, clinical, all the way to bioprocessing, led by BTI, right? So I think, the manpower part, that's largely checked. On the other hand, if you look at the biotech presence in Singapore, it's mainly still dominated by a large pharmaceutical company, their manufacturing outpost, if I may say, in Singapore for clinical manufacturing and mostly commercial manufacturing. So I think we do have a critical mass of people who are really familiar, comfortable with monoclonal antibody production, process development altogether. I guess it's just a little patience that we actually take a leap of faith together out and to say that, hey, this is actually a bright new land that we want to conquer, and put a Singapore flag in there to benefit not just our country, our region, and also the patients worldwide. Why don't we think, innovatively how we can quickly train up the human capability, right? So we have the MAP folks to tap upon. We have solution providers, as Mingming mentioned, the Cytiva training course. And similarly, on the Paul side, we have our own training courses. We also have process development services to help the customers, the drug developers, to jumpstart this capability. So I think it also takes a change of our mindset. So the suppliers like us are no longer a seller where we send our technologies in a
Right.
box and just say bye-bye to you after signing off a PM contract. It should be taking a much more strengthened partnership so that we walk together as early as possible. We frequently exchange in terms of thoughts, the market outlook, and also our deep technology insight, right? So from there to help strengthen each other to build up the manufacturing R&D capability in Singapore, and so that we can benefit the cell and gene therapy community in Singapore altogether.
So we have one question from I know we're running a bit over, so maybe I'll just ask this one last question from our audience. So the question is about how can either Paul or Cytiva maybe help a company overcome licensing regulations within the country? Say like PT you mentioned, like HeLa cells, whether you can bring it or not. So if a company wants to work with HeLa or any other cell lines, but they can't actually obtain these cell lines for some reason in the country due to licensing issues, is this something that either of you would have ability to help a company with such of these problems?
I will share my thoughts first. So definitely, as a business, we have to understand what we can totally own and control, and versus things that we can help provide consultation, that we can help share our knowledge in a more open manner. So I think the cell line licensing, this is a classical example of the latter. As you know, up to now, we don't own this cell line, so we're not really in a position to directly work on behalf of a customer unless this is worked out through a dedicated commercial agreement or something, right? Alternatively, we can also look at different ways. So I'm actually just thinking out loud, right? So if we are actually undertaking a bigger joint effort, for example, a process development as a service, right? If the customer decides to use a cell line that is totally owned, controlled by Paul, for example, then this will be a different matter. Right?
I think Pei-Ching has already covered the cell line part. I also see that there's a reagent issue in the question as well. So in terms of reagents, I'll probably cover the reagent part because we have just signed up a partnership agreement with Nucleus in the US that we are able to manufacture custom-made reagents for different type of cell lines, and that can scale from less than 100 mils or 20 to 100 mils to, even larger, 500 liters. So that would be, if we're able to produce those reagents as required by these companies, and then we can actually ship it over, and then you can do a validation test of which one is actually a comparable product to the one you are trying to license. And then, the IP will still goes back to the customer, that because based on our partnership agreements, we don't hold the license of those culture ingredients, because that eventually will be still be the company's assets, our customers' assets. Although we do manufacturing, but we do have those CDAs in place to ensure that those technologies still belongs to the customers. So that's one of the potential ways that we can solve this reagent issue.
Right. Thank you so much. We're kind of out of time, but I'm really glad to have both of you here to really share some of your thoughts on the cell and gene therapy space. It's a growing space in Singapore, growing space in Asia. A lot of people are definitely interested. So as for all of our audience, those of you who are really interested to learn more, we did post a link for the cell visit to Cytiva. Go online, book one of the slots, and go learn more. Also, you can also reach out to us, and we will help connect you with our speakers if you want to find out more from them. And I believe some of you might have taken down their emails. And with that, we thank you for joining us today. We hope to see you at our next webinar. Thank you, everyone.
Thanks, everyone.
Thank you.
Have a good weekend.
Thank you.